Welcome everyone. This is the last part of the Treatabolome workshop, and it concerns how we are currently building the Treatabolome database for clinicians to submit or access disease gene treatment information. So I am Leslie Matalonga. I work at the SINAC ESHG in Barcelona in the bioinformatics unit led by Sergi Beltran. So the concept and the rationale behind the Treatabolome have been explained in the first part of the workshop by Giselle Bon, and here we just want to remind the need behind the development of the Treatabolome database. So as you may know, over 7% of the population is affected by one of the approximately 8,000 rare diseases, and 80% of those are of genetic origin, and this is why it's important to identify the disease-causing variant for providing a diagnosis. However, these disorders are in general clinically and genetically very heterogeneous, leading to what is called the diagnostic odyssey for the patient that might take on average almost 5 years to reach a final diagnosis for them. So this diagnosis is of course crucial for accessing proper treatments, which could be useful to prevent the progression of the disease, to help on managing symptoms, and also, of course, in less number of cases, but they also exist, curative treatments. So fortunately, with the next generation sequencing era, genetic sequencing has become readily available, and patients with rare diseases may be diagnosed by physicians who are not specialists on specific disorders and might not be familiar with the treatments available for those. Therefore, to avoid a second odyssey for the patients to get access to treatment, clinicians would really benefit from accessing disease gene treatment databases such as the proposed Treatabolome database.
The core of the Treatabolome database is the information that has been entered by disease experts into our system. There are two main ways of entering this data: through the systematic expert reviews, and I won't enter into more details for that, as Rachel Thompson in the second part of the workshop has already discussed that. And also, the data can be entered by individual submission based on publications. So the data that has been entered in the system is made accessible to clinicians through a user-friendly interface that we will show with a short video demo at the end of the presentation. And also the information from the database is made accessible to entire systems such as the RDConnect Genome-Phenome Analysis platform through programmatic access. So during this part of the workshop, we will walk you through this data entering process and show you which information is collected to enable flexible queries on the system for clinicians to access and take medical decisions.
The core of the Treatabolome database is the information entered by disease experts into the system. There are two main ways of entering this data: through systematic expert reviews, and I won't enter into details for that as this has been explained by Rachel Thompson in the second part of the workshop, but also data can be entered by individual submission based on publications. So once the data is entered in the Treatabolome database, is made accessible to clinicians through a user-friendly interface that we will show later on with a short video demo in this presentation. And also the data will be accessible to other database and analysis systems such as the RDConnect Genome-Phenome Analysis platform to integrate this information through programmatic access. And we will also see an example of that. So during this part of the workshop, the last part of the workshop, we will walk you through this data entering process and to show how the information is collected to enable flexible queries on the system for clinicians to access and take medical decisions upon that information.
The database is based on collecting information at four main levels: the disease, the variant or gene, the treatment, and the publication. The inclusion criteria for an entry to be created on the system is to report a treatment at the gene or gene variant levels that have been published and peer-reviewed in English. The dimension of the population, if it's a case, a series, a cohort, the timeline of the study, if it's a retrospective or a prospective study, the existence of comparator or randomizations are not exclusive to enter the database. However, outcomes, qualitative or quantitative, should be mentioned with the intervention.
The core of the data is based on collecting information at four main different levels: the disease, the gene and or the variant, the treatment, and the publication. Inclusion criteria for an entry to be accepted and created in the database is to report a treatment at gene or gene variant levels that has been published and peer-reviewed in English. The dimension of the population, if it's a single case, a series, a cohort, or other study population. The timeline of the study, if it's retrospective or prospective. The existence of a comparator or randomizations are not exclusive to enter the database, but outcomes, qualitative or quantitative, should be mentioned with the intervention. Cases regarding generic therapeutic intervention, for example, benefiting from physiotherapy for a diverse group of patients, would be excluded from the database. Therefore, if a study meets the inclusion criteria, it can be easily entered in the Treatabolome system through a user-friendly form that has been developed. The process will walk the users to different steps, mainly concerning the four levels that we mentioned before. So, information about the disease, the gene and or the variant, the treatment associated, and the publication where the study belongs. So the first step, which as mentioned is an exclusion criteria if the data has not been peer-reviewed, is to enter the PubMed ID of the corresponding study. By entering this PubMed ID, the system will automatically retrieve all the publication information, such as the title, the journal, authors, etc. And in the future, we also plan to expand this data entry with other databases such as clinical trials, Central, WHO, or Eldracity. The second step concerns the description of the clinical information that will be associated to that gene variant treatment entry. So a disease level description is mandatory, and we also enable other granularities such as the specific molecular diagnosis using the OMIM ontology and the symptoms using the HP ontology. The next step will be to also integrate the MAX ontology at different levels, and this has been explained in details by Peter Robinson in the previous talk. So this structure ontology based will enable flexibility when identifying mutation and gene-dependent treatments on phenotypically heterogeneous disorders. And as we will see later on, this will really help to do different types of query and answer different use cases. The next step is to enter all the variants reported in the study, and the system will check that the correct nomenclature has been used through variant validator. So once the different variants have been entered in the system, we also enable to describe and insert specific genotypes, as in many cases, such as for reporting treatment response on autosomal recessive cases, the treatment will be associated to a specific genotype involving different variants. The treatment is then defined as the combination of a drug or a cocktail and a variant or a genotype. We plan to use the Chevi ontology to validate and classify the drugs. And if several drugs have been administered together, this is also captured in the database, enabling to generate different treatment combinations, such as a drug to variant, a drug to genotype, cocktail to variant, or cocktail to genotype. Finally, the last step is to review and confirm that the data entered is correct. Once reviewed, the entry is submitted to the system and stored in the Treatabolome database. Here, I don't want to enter into technical details, but the Treatabolome database is a PostgreSQL relational database composed by 13 tables that enables a large flexibility among connection at different levels, which will be very useful to answer queries from different use cases and different entry points, such as queries at the gene level, variant level, disease level, symptom level, etc. So this is the current user interface. We are still working on it to provide all the information and granularity available in the database. In the left panel, we have the filtering parameters for now by gene or publication number, and the white part corresponds to the result section. So if we zoom into the result section, you can see that on the left, the results correspond to a list of genes, which indeed is taken from the CMS review published by Rachel Thompson and collaborators, and that has been mentioned in the second part of the workshop. So there you have the list of the genes associated with their corresponding treatment, the grade of response, currently low, moderate, high, and adverse, but we are still working on better defining these grades and associating scores. And you also have the number of publications that support that specific association between the gene and the treatment. Then, if you select one of the genes on the right panel, the different publications with the number of cases participating in each of the studies, the year and the type of case will be displayed. In the top bar, you will have a summary with the first line and supplementary treatment for the gene selected and the key publications supporting those results. We are now working on adding variant and specific clinical information on top of all these results. Here is a short video on how to navigate through the database. Finally, as I mentioned at the beginning of the presentation, the Treatabolome database will have the means to provide programmatic access. Therefore, analysis tools such as the RDConnect Genome Phenome Analysis platform, which currently have more than 13,000 rare disease datasets, will be able to flag the variant that have been associated to a specific treatment in the Treatabolome database. This will accelerate treatment identification when identifying the disease-causing variant in the system. Therefore, clinicians will identify the molecular diagnosis and know the treatment options simultaneously, and this will really accelerate patient access to those treatments. Here in this screenshot, you can see a list of variants, the first one being the causative one for the disorder, and how the system flags that variant as associated to a treatment by Clicking there, then the clinician would go directly to the Treatabolome database to have more information for that specific treatment and variant associations. Before finishing, I would like to acknowledge the SYNACT CRG, the Treatabolome database technical development has been done there in Barcelona in the bioinformatics unit, which is led by Sergi Beltran. So thanks to David Piscia and his engineers team, especially to Carla Hernandez, who has developed the Postgres database, and to Alberto Corvo, who's working on the front-end interface. Thank you all for your attention, and please feel free to contact us if you have any questions. The emails from Sergi and myself are in this slide. Thank you.